All right. Next up online is Hannah from MIT. Hannah, are you online?
I am. Can you guys hear me?
Yes.
All right, and you can see the slides.
Yeah, we can see your screen.
Wonderful.
I can't see you though. Yet.
Oh, it should be fine. It'll mostly just be the slides here.
Okay.
All right. My name is Hannah Diehl, and I'm a graduate student in the Electrical Engineering and Computer Science department at MIT. And on behalf of our collaboration with Novartis, I'm here today to present our C-Value app, a visual decision-making tool for drug development. So to understand how this tool might be useful, I'm going to describe a scenario. Let's say you're looking for proof of action for some new drug, and so you decide to look at how over 5,000 relevant biomarkers respond to this drug at different concentrations. You run some linear regression and then pick the top 5 biomarkers with the strongest response. You think it looks pretty good. You see some pretty convincing plots here. You even run some standard statistics and find that you get some pretty good looking p-values. Based on that, you might decide to invest a lot more time and resources into investigating this drug, but ultimately it would reveal no effect. In fact, all these plots just came from synthetic data where I just generated the exposure and the concentration to be independent. The only reason you're seeing results that look so extreme is because you ran that linear regression 5,000 times. At least 5 of those times, you'll get kind of extreme looking results. So the question is, what went wrong? Why did we look at this, feel so compelled by our plots, and end up investing in the wrong drug? In pharmacology, the signals that we see in exploratory data analysis inform trial design, and this involves a lot of investment of time, money, and a lot of opportunity costs. But the problem with relying on these signals that we see is that humans are really good at seeing signals even when they aren't there. This is why, forever humans have looked up at the stars and could say they saw people and archers, and that's why if we look at this nebula, it ends up getting the name Horsehead Nebula. But these things were just totally random, but as humans, we look at it and think there's meaning there. And even when we're aware of this fact, it's really hard to avoid. So what we wanted to do was design a visual check for significance that's fast, easy, and intuitive, and is convincing even when you are otherwise incentivized to see a signal. We decided to do this by creating an R-Shiny app that could be used by anyone on the web for their own analysis. So we've contributed the C-Value app. It's an R-Shiny app that facilitates visual permutation tests that have been described by Booya, Majumdar, Raichaudhuri, and others. We designed it to support a number of common inference tasks used in drug development, and we paid a lot of attention to how we can enable some team-based voting and significant calculations so you can get multiple people involved in this process. This is kind of what the landing page for that app looks like, and you can visit it live now at www.cvalue.org with a dash. So the heart of this application is the lineup protocol. To explain the lineup protocol, I'll actually just run you guys through a quick test of one. So each of these plots show some Kaplan-Meier curves for 3 different strata of participants. And I just want you to look at these plots and decide just to yourself which one looks the most different from the others. You don't even necessarily have to know what you think is making it distinct. Just try to figure out which facet draws your eye. For me, it's this plot 19 down here. Mostly, I notice the difference in the fact that the strata 0, 1, and 2 are more well separated in this plot than in any other plot. And that's exactly how you do a lineup test. We basically start with a hypothesis that strata influences survival. Then this inspires the idea of a permutation test. That is, if the strata has no influence, then if I scramble the strata label, it shouldn't really change the plot appearance. So just using those 2 principles alone, we developed the lineup test where we plot the real data along with these different copies of different versions of scrambled data. You then instruct the user to pick the most unusual plot. And in fact, you can leave this prompt very open-ended, and this allows users to pick up on kind of any signal without you having to know a priori what that signal is supposed to look like. Then the idea is if you and your teammates can reliably pick your real data out of the lineup, the result may be significant. That means something is making it distinguishable from this data that was otherwise generated under the null hypothesis. So to then just explain how you would use the app that we've designed, I'll walk through just a single workflow of team-based voting. So the first thing you do is select your analysis from our menu of supported analyses. We support subgroup analysis, exposure response analysis with options growing. We can look at responses that are binary outcomes, continuous outcomes, or survival. And it's been designed so that it's pretty easy to add new analyses into the pipeline here. Once you've selected analysis, you can explore a preloaded vignette, some sample data that'll then just give you a lineup so you can practice with the lineup and voting procedures. And then once you're ready, you can upload and configure your analysis. All you have to do is upload your data and just tell the app which column names correspond to like what meanings for different variables. You can pick some plot settings, and then there's an option to download the analysis setup that you can then share with your team. Once your teammates get that, all they do is upload it, and it'll, without having to then further configure anything, it'll show them a lineup, and they will be prompted to vote on which facet they think has the real data. And they'll be given some immediate feedback on whether they were right or not, and they can report this back to you as the team leader. Finally, once everyone in the team has voted, you can use the in-app calculator to figure out a C value. You just tell us how many people performed the lineup test and how many got it correct, and it'll calculate this value. This has an interpretation pretty similar to a traditional p-value in the sense that what you're looking for is, depending on your threshold, you'll set a value that you'll set a threshold that if you get a C value lower than that, you'll consider it a significant result. Basically, what the C value is doing is if there is some chance that, even if the plots were fully indistinguishable, some people are just going to randomly vote for the correct plot. So it basically will tell you the probability that people just happen to all vote for the correct plot, even when there was no real distinguishing feature. So go ahead, use our app. It's live already. Avoid tricking yourself into thinking you have significant results when you don't. The app is still actively under development, so if you have interest in the app or requests, questions, reach out to me at hdiehl@mit.edu. And I just want to say thanks to my collaborators, Andy Stein and Naladri Roy Chowdhury at Novartis, and my advisor, Tamara Broderick. And thank you for your time.
Yeah, thank you very much, Hannah. That was a great talk.